Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 170th seminar, and I call it a Georgia Tech Day. I just came back from Atlanta where I attended a workshop to give career advice to future leaders of scientists and engineers. Despite all the difficulties and tough situations in research communities, I observe promising and passionate next generations of researchers coming back home with hope and optimism. Thanks to those people, the future is much brighter. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Mark Styczynski. Mark is a professor at Georgia Tech, coincidentally in Atlanta, from which I just came back yesterday. More importantly, he was my numerical method and computation course TA when I was a graduate student at MIT. So I have known him for 20 years, literally, and he is a true pioneer who changed his research field to synthetic biology because he was mostly, computational person, I believe, at the MIT when he was a graduate student. So, Mark, thanks so much for your amazing contributions to the research community, also leadership in EBRC, the Engineering Biology Research Consortium, and your service is very valuable to all of us. And the virtual podium is all yours now. And thank you again.
Awesome. Thank you. Is that sharing properly?
Yes.
Cool. Yeah, so thank you so much for the invitation. It's cool to be here. And I'm gonna give a kind of speed run through a kind of swath of work that we do. We do a bunch of diverse things. And as was just said, I came from a computational background. I've landed in a very different space. And it's stuff that's really exciting to me and rewarding to me, and I love to get to share stuff with people whenever I can. And 5 to 10 minutes is not enough to actually tell any real science, so I can just do this super high level overview that I usually don't do. But first, I'll tell you why we do what we do, because we think about what it takes and what happens when you need to get diagnostic testing done. So unless you're taking a COVID test, you're likely doing some kind of blood test. And often that entails going to some big, fancy, expensive, potentially scary hospital covered in glass. And when you get there, they stick a big needle in you and pull out a bunch of blood. That stuff gets sent up to some really fancy equipment that's going to do some kind of magic to it. And then after it produces its magic, you get the results, and then you also get a big fat bill, right? And essentially, all levels of this are suboptimal. And what we're trying to do is change as much of that as possible. We want to have tests that can be done through a finger stick rather than needing an entire venous blood draw with a big old needle. We want to have things that can be done for pennies on the dollar. And we want them that they can be done essentially anywhere. So you don't need to go to some big glass building. You could do it someplace where there might not even be reliable electricity. And so then the way we do that is synthetic biology. We harness nature, we harness bacteria for us in order to create sensors that can be used as diagnostics. And the specific thing that we've spent a lot of time doing lately is cell-free protein synthesis. Basically, we take bacteria, we grow them up, we bust them open, and what's left is this rich reaction mixture that when you purify and process it, you can do a lot of cool things with it. You can create customized synthetic biology-based sensors that can detect biomarkers of interest. And so what that essentially means is that cell that you had just busted open, that's essentially your mass spec. That's your big fancy instrument that otherwise you'd be using, but now you can use it in a much smaller version and a much cheaper version. You can do a classic fluorescent reporter that everybody's synthetic biology, everybody and their uncle in synthetic biology is familiar with how to use fluorescent reporters. But then we can also use colorimetric reporters so that we can get things that you don't necessarily need equipment to read out. And you can imagine that that allows for better field deployability. And so that's kind of the underlying approach that we use, because our goal is ultimately to make things that are essentially like pH paper, except can it be for maybe everything else in the world in addition to pH? We want it to be as easy to use as that, but hopefully a lot more modular and generalizable. And so just an overview of a couple of things that we've done. Kind of our starting point was just making a zinc sensor because zinc deficiency is a global problem in terms of nutritional deficiencies. And so Monica McNerney was able to get us involved in cell-free systems. And she made a zinc sensor where you can get a colorimetric readout in 50, 60, 70 minutes that you can read with the naked eye. And there's other kind of finer details that we dealt with and further advanced how to make these more fully quantitative and control for the effects of different people, different patients' samples. But that's not stuff that we really have time to get into. But this color change can be used as essentially a pH test for zinc deficiency. And so, what other cool things do we think we could do with it? Well, Yen Zhang, who's another graduate student in my group, she said, "Okay, can we take these cell-free systems?" And there's this guy down the hall, Shu Takayama, who's an amazing guy, and he does these things called aqueous two-phase systems. Now, you might be familiar. Oil and water don't mix; you get two phases. There's actually some polymers that can be both in water aqueous phases, and they still don't mix. And that's what Shu does. And we combine that with what we do to make these kinds of protocells, where if you look at from above, in one small, tiny little well, you can have multiple different droplets in one of those phases that have different biosensors. And then your overall well, separate from those droplets, would be where you add your sample. And so the idea would be that you can load in a sample and you have all these different sensors. And then if your sample has target A and not target C, then sensor A is going to light up. And we showed that we can do that. We can have a little well, and this is no bigger than 100 microliters, and you can add in your sample, and you can get different signal. And we can even lyophilize this stuff so that you can imagine that makes it more field deployable, and we can get individual responses. Here is an example of us sensing DNA and RNA. We can do it for small molecules. We can do it for ions. Yen also did some super cool stuff saying, hey, well, we do these things that we call semi-quantitative because it's a visually interpretable output. Is there any other way that we can do things that are still super cheap, but maybe more quantitative? And she said, well, maybe I can use a glucose monitor. That's something that's out there that everybody can use. It's reliable, it's cheap. Can we make it so that it's not about us measuring the glucose in blood, but maybe we can take our sample and based on how much of our target there is, produce a bunch of glucose that then the glucose monitor can measure. And she showed that we can do that. So we can, as usual, we go to our go-to detecting zinc in water, and then also detecting zinc in serum. So we showed that this can really be used to have a portable glucose monitor. It's a cheap and quantitative way to measure things. And one of our most recent things that we've done is enabling protein detection at the point of care. So if any of you have ever had to measure proteins in the lab, you've maybe done an ELISA. If you're familiar with it, you probably think that it's miserable because it takes forever. And this actually is science. It always stuns me that this is part of a scientific protocol that you're like throwing stuff practically into a trash can. And you do this a bunch of times with a bunch of incubations, and then you try to get your results out. It's complex, it's laborious, it's time consuming. So if you want to do the point of care, that doesn't work. But what people do at the point of care are LFAs, that's your COVID test. And these are potentially great, but they often lack sensitivity. And most critically, they're not particularly modular. It took us a year with the world having almost nothing else to do to get a COVID lateral flow assay, right? So is there something that can be more modular, more quickly deployed, and give you maybe more quantitative results? And Meg McSweeney, another grad student in my group, was able to take a split T7 polymerase where you take an RNA polymerase, you split it in half. Somebody's already done this for us. We love the Dickens Lab for doing this. And you fuse to it essentially some antibody fragments, some nanobodies. And when you have your antigen there, both of these guys come and bind to your antigen. The polymerase reassembles, and then you can do transcription translation. We call it T-LISA because it's T7 RNA polymerase version of ELISA and make sure that we could do this. Take a standard synthetic biology approach, and we can get our same visually interpretable sensors. And we can do this in any kinds of matrices. You can do this in a serum or a saliva sample, and you can do it for model proteins, or you can do it for real proteins that you actually might want to detect, like the SARS-CoV-2 RBD. So again, that was kind of my speed run through the stuff that we do. We try to sense anything, and we try to do it in a cheap and deployable way. And I've been very fortunate to work with some amazing people. And I'll kind of leave off with some unsolicited advice about kind of how I got here and how I think that I've found what I think is a happy place for me. The first is advice that I got told when I was uncertain about what I wanted to do. A guy that I trusted told me, follow your Zen, right? Which is ultimately like a Phil Jackson thing. But don't worry about whether you can or can't do it, right? If you think it's exciting, if you think it's going to fulfill you, then go for it, right? We're all fortunate enough that we're well educated and likely to be really successful, such that even if we fail initially, whatever we fail into is still probably going to be a productive, safe life for us. Be receptive to new ideas and opportunities. As Taysok mentioned, I was a computational person and then I was a metabolomics person. And then 10 years ago, this opportunity arose and it turned into two-thirds of my lab. And it really helps me feel fulfilled about what I could potentially do for society. So be receptive to these new ideas, be open to them, and try and have self-confidence, right? There's a lot of people that wonder, can I make it? I can't do it. I'm not smart enough. They sometimes look at some other people's self-confidence and they might have a lot. And then that makes them wonder about themselves. So please have confidence in yourself. You got here, you are doing a great job, and don't worry about what other people say about themselves. And then finally, elevate those around you both by praising them and pushing them. Because yes, telling people what they do right is exactly what you need to do, and telling them that they're amazing and doing great things is exactly what you need to do. But you also need to find the ways to help them become better, to self-actualize. So push them, question them in a positive way, in a friendly way, and help them to become the best possible versions of themselves. So that's my random unsolicited advice. And I'll shut up now. And thank you again for getting the opportunity to chat.
Thank you so much. I mean, you know what? Yesterday and the 2 day for 2 days, I was in workshop, basically giving advice. You beautifully summarized what I did, what I did say yesterday and 2 days. Uh, Also, especially, I just wanted to add one more thing. I sometimes ask question to young people, what's next? What's next in your career? And I realized I need to change my question a little bit after multiple interaction. What makes you happy?
Mm,
Probably that's a better question because I mean I realize what's next is a very stressful question
mm,
to those
mm,
people because they start thinking about this one for many years. But the main goal of our life, I believe, is pursuit of happiness. But other thing is basically helping that goal. So getting a job is not actually the becoming happier or
mm,
happy. So I now somehow realize, and I believe Mark is one of the best advisor in the world. And based on my interaction with his advisee, including he already mentioned Yan Zhang, she also was the rising star speaker in this series. She is super bright, super, excellent, and that is thanks to him, I believe. Thank you so much.
thank you.
Yeah. All right. Now, the main speaker of today with a longer introduction. Professor Nils Averesch is a assistant professor at the Department of Microbiology and Cell Science, University of Florida. Before joining it, he was a research engineer at the Department of Civil and Environmental Engineering, Stanford University, and an associate scientist at NASA Ames Research Center, Space Science and Astrobiology Division. As you can see, I mean, he was all of the different field, and he also basically following Mark's advice, I guess, already. Nils holds a PhD in metabolic engineering from the University of Queensland, Australia, and also an engineer's degree in biochemical engineering from the Technical University of Dortmund, Germany. So geographically also he was all over the place. The research of his lab comprised the rational engineering of microbial metabolism to increase the carbon efficiency of biochemical pathways for the assimilation of single carbon compounds and the conversion into advanced biomaterials. This serves the overarching goal to create a sustainable chemical industry on Earth on the way to new frontiers. In other words, developing circular production platforms based on microbial biotechnology, I believe that could one day also support crewed long duration space exploration missions and settlement, hopefully in Mars or Moon. He and I share similar goals and missions of space exploration, leveraging synthetic biology, and I hope his effort will be successful so that we can someday visit Mars or Moon. Nils, thanks so much for your time today, and please take it away. All yours now.
Thanks so much for the introduction, Tae-Sok, and thanks for having me. Let me see how to share my screen, the right one. Does that look good?
Yep.
Cool. So yeah, welcome everyone to my talk on microbial cell factories for space and Earth. Where I'll talk about how to turn single carbon feedstocks to advanced biological plastics. And yeah, I'm with the Department of Microbiology and Cell Science at the University of Florida. And the University of Florida its main campus is in Gainesville, but I'm not in Gainesville. I'm at the Space Life Science Laboratory, which is just outside the main gate of Kennedy Space Center. And yeah, that makes a lot of sense for the research that I do, as you will learn on the following slides and throughout my talk. So what are the resource constraints of human spaceflight? Basically, human space exploration is and always has been reliant on resupply. And launching stuff to space is expensive. It becomes cheaper now with more and more different launch providers coming up and new reusable rockets being developed. But ultimately, it will always be expensive. And if you look at the destinations that are possible, that multiplies the cost of these missions. For example, going to the moon, you can get there relatively quickly. And then you can spend a number of time on the moon. But basically, there's always a mission abort scenario because the moon doesn't leave Earth, really. So whenever something goes wrong, there's still a chance, an option to go back. That changes with other destinations, such as asteroids or Mars. While the journey is significantly longer to get, for example, to Mars, this is also a trip that you can't cancel. Once you're on the trajectory, you're going. So that dictates the resources that you have available and what you need to bring with you. So not only the carry-along capacity is very limited, but you also have to plan for every eventuality because there's no mission abort. And that makes it really, really difficult to plan what to take. And ultimately, in situ manufacturing will be imperative for these constraints, for these reasons. But yeah, what resources can we actually rely on other than what we bring? Well, in space, really en route to some destination, there's really only sunlight and the cargo, what you bring with yourself. Stuff that you might have on station in your spacecraft. Going to the moon, you have some rock regolith, which is not super useful. There might be some water to be found somewhere, but that is really difficult to mine. Depends on the destination, on the landing zone as well. Going to Mars, though, there is an atmosphere, and that is fortunately composed of carbon dioxide, for the most part, 95%, plus some nitrogen. And that could theoretically enable in situ resource utilization, is what it's called, using biotechnology, because carbon, nitrogen, and water that also exists on Mars in reservoirs underground or at the poles. Those are the main compounds that biology uses to basically to grow and to replicate. So that creates the opportunity for biotechnology for space exploration. Specifically, the further away you go, the more imperative, but also the more useful it becomes. So what are the resources that we should be focusing on? Well, I usually categorize these into four classes: food, pharmaceuticals, and materials. And they have a different type of demand and volume that dictates how these should be made or brought. So food has a very regular demand to keep astronauts healthy, and a very consistent volume, rather predictable diversity as well. Pharmaceuticals, you can't really tell when you would need these, but if you need them probably immediately, although only small volumes. And there's a wide variety. So the question is, what can we make? What can we just pack up? Materials might have incidental demand, for example, for replacement parts, if gears break or things like that. But when you need that, a gear is a bit more mass than a pharmaceutical, so you need a larger volume. Applications are limited though, but yeah, as I said, the volumes are rather large, or can be rather large for depending on what breaks. So that basically dictates what kind of biotechnological approach we could take to convert in situ resources like carbon dioxide or derived single carbon compounds, feedstocks into these supplies. And this is part of a white paper that we wrote a couple of years ago for NASA, where we basically looked at what kind of conversion pathways and strategies and alternatives exist and how they tie to the final products that might be needed in space. So at the beginning, there's the carbon conversion strategy, and then the carbon feedstock class, which can be just a gas or liquids or complex compounds like biomass. And then from these basic carbon feedstocks, specific chassis organisms are useful to convert these into the final product. And what chassis you choose depends on, what substrates these can use, how fast they are, probably also how genetically tractable these are, because that'll all dictate how to scale the process and what volumes and rate you need your products at, and titers, purity as well. So here the ideal path really depends on various factors, like I said, and multiple runs through this are also potentially possible. So you could, for example, think about recycling stuff that comes out of the astronauts or other dead end products like packaging materials. And that's what we then thought about, basically a concept of operations for supporting a base on Mars. This can also be translated to some degree for lunar application, but specifically here, this is for Mars. So it'll always start with some pre-deployment cargo, which will support a crew and some kind of habitation. Then loop closure can support this so that resupply becomes less important and the mission would be more resilient. Basically recycling whatever the astronauts produce. This may be supported with in situ resource utilization, which I talked about earlier. So atmosphere, water, potentially even regolith being processed and used to support whatever you lose due to the second law of thermodynamics, to feed substrates back in. Ultimately, if both are connected, that should enable autonomous operation of a extraterrestrial base, of an off-world base, making the habitat independent and autonomous, independent from resupply. It's the ultimate goal. Yeah, and for this, basically the key is to convert single carbon compounds, because ultimately everything breaks down to carbon dioxide, and carbon dioxide is also what the atmosphere of Mars contains. And specifically microbes excel at decomposing everything down to carbon dioxide, but also at utilizing carbon dioxide. So an integrated loop closure and in situ resource utilization biomanufacturing for biological life support will, I believe, be based on microbial biotechnology. And yeah, this basically exemplifies how I think this should work on a more like systems metabolic level, where all the carbon dioxide that is produced by biology gets recycled again through microbes, and external carbon dioxide can be used as a support function to create and potentially ultimately scale a biomanufacturing of Earth. The other thing that drives my research is the need for more sustainable materials here on Earth. So this picture that I took a couple of years back on a vacation in Cambodia really exemplifies the problem. There are also there's more numbers behind that can exemplify the problem on a larger global scale. So here, the plastic emissions for the next couple of years have been predicted based on how we try to mitigate our plastics waste production. And even with an ambitious goal to reduce plastic waste, plastics will continue to accumulate in the environment. That's the blue cone here in the middle. So what we really need is to not just mitigate the waste that we release into the environment, but also to find new alternatives for more sustainable synthetic materials, plastic materials. If we don't want the total weight of plastic in the oceans to exceed the total weight of fish in the oceans, which is predicted to happen by 2050 if we keep going as currently. So to avoid that, I'm working on enabling the production of advanced biopolyesters. And specifically, there's one microbe that I really think has great potential for this, which is Cupriavitis necator, H16. It is lithoautotrophic, and it can utilize carbon dioxide as a sole carbon source using reducing equivalents from hydrogen to assimilate carbon dioxide and synthesize biomass. It is much more versatile in its substrate spectrum, though. It is mixotrophic and uses a range of different carbon sources, often in parallel, including volatile fatty acids, also some sugars, making it really useful for upcycling of a range of waste, not only carbon dioxide, but also volatile fatty acid that might come out of anaerobic digesters and processes like that break combined organic waste streams down. And Cupriavitis necator also accumulates polyhydroxybutyrate, which is a biological polyester. It accumulates in granules inside the cell. Here's a formula of the structure of P-poly-3-hydroxybutyrate, P3HP. And when extracted from the microbes, it forms a resin that already looks a lot like plastic. Here's a picture from samples from our lab. And yeah, these polyhydroxyalkanoids, class of natural biological polyesters, can be obtained in a one-step bioprocess from single carbon feedstocks and other waste streams. And they are, to some degree, 3D printable, moldable, and thermoplastic. This material is really similar to polylactic acid, which many might know from the single-use plastics that are taking more and more over in certain industries now, in certain areas. Mostly we know this probably because it breaks really quickly in your hand when you try and have your fries or something that's a bit stiffer. Yeah, that is the same problem for polyhydroxybutyrate. Both have a really rather low Young's modulus, which defines the elasticity and the low elongation at break. So these materials are really brittle, making them not very versatile for a lot of applications. Plastic cutlery works to some degree. Poly-3-hydroxybutyrate has the problem that its melting temperature is close to its decomposition temperature as well. So it's hard to process. That's basically why polylactic acid has made it, because it has a rather high decomposition temperature as compared to the melting point. So that it's easy to process. It's not a great material, but you can process it. And that matters for these industry applications. So the question is, how can we make PHB, the natural polyhydroxybutyrate, better in a way that it becomes a more versatile material? Well, what we did is we changed the enzymes that synthesize these polymers, that take the monomers and chain them together to become a polymer. And we used two different PHA synthases. There's different classes. We used the class 1 and the class 2 and expressed this in cupriables negator, which already makes PHAs. And then by feeding levulinic acid as a sole substrate, we were able to tune the composition of this polyester to not just contain poly-3-hydroxybutyrate, but also poly-3-hydroxyvalerate and poly-4-hydroxyvalerate. So when feeding levulinic acid to the wild type, we're getting a polymer yield of around 6%. And depending on what other mutant polymerase PHA synthase we use, instead of the natural one, we're getting a similar yield. The FASI NSDG, about a comparable yield. The FASI 1310 gives a little bit less polymer. What is really interesting, though, is how these impact the composition of the polymer. So I'd like to draw your attention to the figure here on the right, firstly. The first bar in this chart, where it says H16, that is if you feed levulinic acid to cupriables negator wild type, the polymer is composed of almost 50-50 poly-3-hydroxybutyrate and poly-3-hydroxyvalerate, which makes the polymer softer and lowers the melting temperature. We saw a similar thing with the mutant PHA synthase FASI 1310. And when overexpressing that here on the right side of the split bar graph, the bar in the middle where it says induced, we're getting a much higher incorporation ratio of 3-hydroxyvalerate, around 80%. And that really changes the material. Now it's more waxy than a brittle resin. And that's really interesting because it means depending on what enzyme we use and how much we overexpress it, we can tune the composition of this polymer from a really stiff and rigid polymer to something that's soft and more rubbery. Very interestingly as well, we saw with FASI NSTG, the bars on the right, so here the third and the sixth one, that we can also incorporate 4-hydroxyvalerate in this polymer, which again nuances the polymer properties, because now we have a polymer that's composed of 3 different monomers. And by expressing these more or less the enzymes, we can modulate that composition. In the bar here on the very right, for example, we have almost equimolar fractions of 3-hydroxybutyrate, 3-hydroxyvalerate, and 4-hydroxyvalerate. And we were even able to obtain a polymer that is almost like a rubbery foil, like really, really ductile. And we're currently in the process of characterizing this material. But where does this come from metabolically? Well, the pathway of levulinic acid catabolism, shown here on the left, proceeds via a couple of steps, including precursors for polyesters. Turns out that the FASI 1310 can only polymerize 3-hydroxyvalerate and 3-hydroxybutyrate. So even though the pathway proceeds via 4-hydroxyvalerate or 4-hydroxyvalerate, that is apparently not incorporated. So it goes straight down and then to the left from 3-hydroxyvalerate through TCA cycle towards natural 3-hydroxybutyrate biosynthesis. While Part C and SDG apparently can also leverage 4-hydroxyvalerate-CoA as a substrate. So now we have here shown, highlighted in blue, 3 substrates that the PHA synthase can utilize, and that's how we end up with a polymer that's composed of these 3 subunits: 3-hydroxybutyrate, 3-hydroxyvalerate, and 4-hydroxyvalerate. And yeah, that's pretty cool. We weren't able only to make a polymer that is tunable in its composition, but we were also able to basically elucidate this pathway that had not yet been described to that degree in Cupriabilis necator. Now I'd like to shift focus slightly to another story, still about the polyhydroxyalkanoates, but a different class of PHA. So first of all, I'd like to give an introduction of some really exciting materials. These are polyarylates. Basically, think of them as of Kevlar. They're not Kevlar. Kevlar contains an amino bond, but they are the same type of liquid crystal polymer and used for really high strength applications. For example, sailcloth is made from materials like that. The one we see here on the left is also a bulk polymer, that is polyethylene terephthalate, which makes up every plastic bottle that contains a fizzy drink, basically, that you know. So it is a really good material, really useful for many applications, not just for high strength, but also for commodity products, packaging. It has even been used, these materials have even been used for exciting applications like solar sails. So the picture here on the right that shows the design for a new satellite that is powered by solar wind, basically. And these sails can be made from these materials. And the table below shows how these materials compare to polylactic acid and polyhydroxybutyrate. So most importantly, the tensile strength is much higher. If you look, for some example, for Econol, the tensile strength there is 125 megapascals. For PET, especially the elongation at break is really good, making it a much more flexible material, much more elastic, while it still has a good tensile strength and good stiffness. So since these are all polyesters, I was thinking, why can't we make these biologically? Like, they're all still derived from petrochemistry, because they contain this aromatic compound that is ultimately derived from benzene. But nature also makes aromatics. So shouldn't it be possible to also make these polymers biologically? So I was thinking, can't we make something like this? Just put an aromatic ring into the backbone of that polyester, which should give us a material that is similar in its properties to these high strength aromatic polyesters, these industrial polyesters. And yeah, that's what I set out to do, basically specifically looking at fluoridic acid, which is a compound that can be derived similarly to levulinic acid from lignocellulose biomass, so it's bioavailable, and use a similar strategy, basically modulated PHA biosynthesis. Here on the right, there's the pathway, the normal pathway leading to formation of poly-3-hydroxybutyrate. And I created a second entry step into that by expressing a relative of the enzyme that I showed before, the FASI 1437, which is also an engineered mutant PHA synthase with a really wide substrate spectrum, which has been shown to incorporate a wide range of aryl aliphatic hydroxyalkanoates, monomers, precursors for polyesters. And I complemented this engineering with a CoA transferase because the PHA synthase takes only CoA activated thioesters of the hydroxy acid, so that that reaction is also needed to convert the hydroxy acid into the thioester with CoA. So again, expressing these HAD-A and FASI 1310 in Copiavis negator, I was able to get PHA reduction. Great, that works. We can get regular PHB. The system is functional. And I was also able to incorporate a range of aliphatic hydroxycarboxylates, including 3-hydroxypropionic acid, 4-hydroxybutyric acid, 6-hydroxyhexanoic acid, which all give materials with exciting other properties. Specifically from 6-hydroxyhexanoate, I got this polymer that contained E3HB, Co4HB, Co6HC. And yeah, this is exciting because the backbone is all straight with no, at least except for 3-hydroxybutyrate, with no side chain. This makes this basically similar to nylon, so a material with a really high flexibility and tensile strength. In addition, I tried to polymerize the aromatics that I previously spoke about. Specifically, I was looking at 5-hydroxy-2-methylfuranoic acid, which I managed to incorporate into that, which was able to be polymerized with that engineering strategy. And I was also able to incorporate fluoridic acid, as I had previously shown. And this basically shows the polymers that we were able to obtain. And this is really the novelty here, showing that these aromatics can be incorporated in the backbone. Here on the left, there's the polymer that contains the fluoridic acid moiety, and below it, polyethylene terephthalate. And this is just to signify how analogous these materials are structurally. On the right, there's the compound with a furanoic ring. Also here, there's an industrial analog, polyethylene furanoate, which also looks rather similar structurally, at least, to the polymer on the top right that we were able to obtain. Incorporation ratio is still low, though, so we were not able to confirm material properties yet, other than predicting these. That is the next step that I'm exploring currently in my research. While this was all still co-feeding experiments, I also wanted to show real significance that we can get to these exciting materials from carbon dioxide. So in the last few slides, I'll show how we set up this bioreactor that is based on a bioelectrochemical system, where we use Copiavis negator's natural capability to use carbon dioxide as its carbon source, with hydrogen and oxygen as donor and acceptor of reducing equivalence. So yeah, like I said, this was a bioelectrochemical system, basically an electrolysis cell that electrolyzed water, and then we sparged carbon dioxide, which allowed us to cultivate Copiavis negator, our engineered strains, having it grow solely on carbon dioxide. And then supplementing the aromatics, we're able to show that we can make these aromatic polyesters also in an autotrophic fashion. Here on the right is how that bioreactor looks like in the lab. Rather simple. Often these bioelectrical systems rely on a membrane. This is just one cell, making the setup really simple. And we also got a fairly decent growth rate and achieved a significant optical density, not like growing that strain on sugar, obviously, but as compared to other anaerobic systems that utilize bioelectric chemistry and microbial electrosynthesis, we achieved a fair density of biomass. Specifically important was here, though, the point of the addition of the precursor, which had to coincide with the depletion of ammonia, because that is what triggers PHA accumulation. So yeah, that is a system that we still are working on. So we're going to replace the inducer system with a constitutive production of the precursor, so that doesn't need to be added anymore. That's ultimately the goal. Yeah, this just shows growth of the cells over time and how much of a cell density we were actually able to obtain using different precursors. Here on the very right, fluoridic acid, we achieved an optical density of 3. While on the one right next to it with phenolactic acid, we achieved an even higher optical density of 6, which is 10, respectively 20 times higher than what we inoculate with. So yeah, these cells really grow on carbon dioxide and hydrogen oxygen and make aromatic PHAs. So ultimately, I'm looking to expand the system towards a diverse range of polyesters, aliphatics as well as aromatics, and combine these in many different ways to get a whole range of exciting materials, from really high strength resins that can be used for exciting applications like space technology, to all the way to rubbery materials, to basically replace as many commodity plastics as we have with more sustainable and bio-derived alternatives that can ultimately or ideally be produced from carbon dioxide, so that no further carbon dioxide is released into the atmosphere, but that we rather rely on what we already have. And yeah, for that, key points to work on are the PHA synthase and the CoA transferase. And that's currently one goal in my research group, to screen a few new and engineer new PHA synthases, as well as do the metabolic engineering to get to these precursors. And with that, I'd like to thank the funding that I've received from NASA, specifically most of my career, and currently a renewed grant, as well as the Australia Space Institute here at the University of Florida, 50 years. And yeah, I'd be happy to take questions after I introduce my still young, but yeah, now already 6 people strong research group. And yeah, thanks for your attention. Happy to take questions.
Thank you so much. Exciting. And then you already achieved a lot, even very short time at your university, but your stuff, previous stuff is also very cool. Let me start a question. So you also touch some of the challenges to ultimately go or do the space exploration. So what would be some breakthrough technology development or some technology we need to achieve to overcome those challenges?
It's a good question. And I get that question frequently. Yeah. In the lab, for me, it is the genetic engineering, the molecular biology, getting the strains engineered. We are trying to do a lot of genetic integrations currently. Previously, what I did was mostly on plasmids. Engineering the genome is difficult. It's so specific to each microbe, what the tricks are, how to do knock-ins, knock-outs. yeah, I know that there's a couple of FROs, focused research organizations, that are looking into that. So that's really cool stuff. I'm really looking forward to that taking flight. The other thing is protein engineering. Like I said, I need better pH synthesis to get to real high aromatics content in my hopefully high performance materials. And that is also a thing where I see a lot of potential currently. With the release of Bolts 2, I'm super excited that I'll still witness in my lifetime the ability to go straight from like I want this reaction to be catalyzed to protein sequence. Yeah, I think we might be getting there. So that would be a significant breakthrough.
I see. So to clarify for the audience who listen to this one, you are saying genetic engineering is challenging right now because we are mostly dealing with the non-model organism, not E. coli or yeast, right?
Yeah, I don't know how much non-model or model Cupreavis negative still is. There's a bunch of people working on it, and protocols are well established. It is still tricky because everybody does their own thing.
Mm-hmm.
Protocols don't always say what the specifics are.
Mm-hmm.
And even if people are really open with their research, it doesn't like a very well formulated protocol doesn't replace seeing it with your own eyes,
Mm-hmm.
how it's done.
But
That
how
is still
about
tricky.
Sure. How about the challenges that is so unique in the space, including microgravity, gamma rays, also resource limited,
Yeah,
situation over there? Because I mean, even if we engineer something here, the physiology of our bacteria or organism, completely, not completely, but very different from the situation on Earth. So that is another challenges we need to overcome. And actually, my group actually done some, but not entire challenges yet.
yeah. You've probably done more work in that area than I have, testing microbes and microgravity.
Uh-huh. But you're
Yeah,
probably
there
thinking
are.
about more than me because you are, your life, your training is all about that one. So you're
Yeah,
probably thinking
yeah. I
more
know that.
about that one than me. So I just want to hear from you.
Yeah. So I still think it's not going to be such a huge problem as
Mm-hmm.
long as you have a little bit of gravity.
Mm-hmm.
So yeah, in microgravity, things are going to behave completely different because you have no convective transport. Um, Bubbles won't rise in a reactor, so aerobic fermentation is really difficult.
Mm-hmm.
Yeast fermentation becomes a foamy mess. It just, yeah, is really difficult. But as soon as you have some gravity, like on the moon or specifically on Mars, I believe most of the rules still apply. It might be slower, but you should be able to run bioreactors that have, in principle, designed for Earth, should be able to run them on Mars. On the moon with 0.1 Gs, it might be more difficult. You might have to change the design. But yeah, there is still some gravity that you can rely on. The radiation, I'm not super convinced that that's really a problem for microbes. Some will just die and others will survive. That doesn't really work for humans that well. But microbes, yeah, are much more resilient in that sense because there's always a large number of the same cell. And yeah, there's a question in how fast the genome might change of a strain that you propagate.
Mm-hmm.
I have not seen strong evidence yet that that is a significant problem. Of course,
Mm-hmm.
that needs to be thought about and verified whenever you have a microbial cell factory, that you make sure that it doesn't drift genetically. But if you basically have the same freezer stock and cold temperatures are basically for free in space, well,
Mm-hmm.
mostly if you shield it from the sun,
Mm-hmm.
then you should have the same genotype over a significant time.
I see. That's good to know. Actually, the that kind of confirm my recent interaction with researcher from UT Austin and also the national lab. They actually done some experiment simulating gamma ray situation in the space, and then bacteria growth pretty much not that much different from the without that gamma ray simulation. So it's kind of pretty, um, exciting. But the one problem is that experiment is very expensive.
Yeah.
And also only done very short period of time because of that, expensive kind of problem.
Yeah.
So we don't know what will be longer term impact, but probably, will be also shield, using whatever physical barrier in terms of the gamma ray thing. So I guess, I mean, there are much brighter future than we probably thinking about right now.
Yeah. Well, the shielding is difficult in space because what you have in space is not necessarily gamma rays. The radiation that you have there is basically fast protons and these HZE, these really heavy ions. It's only a small fraction of the radiation in space, but that's the most dangerous, and that will just go through everything. So you need a much thicker shielding than you would need here. Like for lead, for gamma rays, you can use a little bit of lead, or leaded glass might even do the trick here on Earth, depending on how strong that gamma radiation is. In space, you need protons to shield against protons. Like I always do the analogy: if you have a medicine ball, which would be the example for a lead atom, and throw a ping pong ball at it, it'll bounce off, come right back at you. But if you have a whole bag full of ping pong balls, you throw one in there and it'll stick. So that's why you need water, basically, or anything that's high in protons to shield against those fast protons in space, and a lot of it.
Got it. That makes sense. So, I mean, but at least the data they show is kind of promising. That actually I listened to that talk during the SIMBD meeting a couple of weeks ago. So, I mean, there are some technology I believe, will be developed, to address all the problem, I guess. And hopefully, I mean, in my lifetime, people, if they have some money, I mean, really visiting Mars or Moon and this. I mean, I want to see that happen.
That'd be cool. Yeah.
Sure. All right. Now I see the 11 Central Time, and let me close. Okay. Thank you all for joining and staying today. We'll meet again on August 21 after one week break, actually 2 week break total. Thursday, the same time, the same Zoom link. We'll have Dr. Blake Simmons at Lawrence Berkeley National Lab, also fellow of National Academy of Inventors, and also Professor Ju-hyun Kim at Gyeongbuk National University, Korea. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces only if you wish. Thanks, and I stop recording. Just give me one second.